Let's see if it is gonna work. No. Is this working? Yeah. It doesn't matter. Okay, so good morning, everybody. It has been really interesting to listen at all the talks this morning. It was really, really good because I'm partially going to cover some of these aspects. Although the main goal of this presentation is really to provide you a sort of global perspective of the current state of the art in Alzheimer's disease research, presenting while the current research system is apparently failing to provide new treatments for patients and how to improve the situation, learning from previous mistakes. So the Physicians Committee for Responsible Medicine is a non-profit organization. We deal with preventive medicine a lot. We promote healthy lifestyle, plant-based diets mainly, but we also deal with research and really implementation of new methods to improve medical research in several areas. So let's ask ourselves, is the current Alzheimer's disease research paradigm failing us? We should look at some facts, and some of this information have been already provided by other speakers before me. So as we know, Alzheimer's disease is the most common cause of dementia, with 50–75% of all dementia cases affected by actually Alzheimer's. And Alzheimer's disease cases in the US in particular is expected to almost triple by 2050, with 13.8 million of people possibly affected by dementia and Alzheimer's disease. Unfortunately, this is a very terrible information for sure, but it's just a matter of fact. In the last 10 years, there have been no new drugs for Alzheimer's disease. And currently available drugs can only stabilize symptoms, but temporarily, and not in all patients. They don't seem simply to work for all people. However, they do not also seem to slow disease progression. So what we're looking at is a terrible, staggering clinical translation failure rate. In Alzheimer's disease, as has been reported in this journal published, in this paper published in Alzheimer's Research Therapy in 2014 by Cummings and collaborators, the clinical failure rate for Alzheimer's disease research is 99.6%. What does this mean? It means simply that 99.6% of drugs tested in animals do not work in humans. Just for your reference, this is the abstract of this publication. It's specifically referring to drugs, in particular, I would say, 413 Alzheimer's disease trials performed from 2002 up to 2012. And along these 10 years of time frame, no new drugs have been worked for humans. Another kind of dramatic situation is that several drug companies, thinking either about Pfizer and many others big pharma companies, are actually currently downgrading their efforts to develop new pharmacotherapeutics for Alzheimer's because it seems to be a complete failure. So at the Physicians Committee, we have been trying to understand why. What's going on? Why are we facing this failure rate, especially in Alzheimer's disease? But not just in Alzheimer's, let me tell you. Also in cancer research, diabetes, many other medical research areas. But in Alzheimer's, I would say it's particularly tremendous. So what we know is that in the last decades, animal models have been extensively used to understand the pathological mechanisms, molecular, cellular mechanisms of Alzheimer's disease in particular. We have been seeing and read probably hundreds of publications describing transgenic animal models, inbred mice. Of course, mice is the most used animal species, but there are many other species that have been used. We heard a little bit about pigs and primates and dogs, really many others. However, what we know about these animals is that, okay, it's interesting. They can provide us some insights regarding formation of amyloids, but also the formation of plaques and the neurofibrillary tangles and gliosis. They show signs of synaptic alterations and signs of cognitive impairments. So they're providing, for sure, some insights regarding molecular mechanism of the disease and cellular components. However, what is pretty clear is that animals like mice, transgenic mice, do not actually develop Alzheimer's disease showing the clinical pathological features that are present in human beings. And also, as we can see from this literature, and this is really a sample of the literature that came out, treatments that seem to work in animals have not translated to humans. Additionally, animal models might also generate the so-called false negative data. So a compound that might actually not work in a mouse might potentially actually work in humans, but this compound will never go into preclinical trial phases simply because it failed in mice or in other animal species. Another thing we should start thinking about is the type of Alzheimer's disease we're studying. So we know that there are mainly two types of Alzheimer. One is the typical familial early onset Alzheimer's disease. The other, much more frequent one, covering 95–99% of all Alzheimer's disease cases, is the late onset Alzheimer's disease, which is also called sporadic because it's not directly related to a genetic modifications or genetic mutations or genetic allele or whatever. However, the majority of the transgenic animal models that are available for Alzheimer's disease are actually covering and might be useful to study the familial Alzheimer's disease, as we have described in this publication from our organization. This is a table. I'm not expecting to go through all these details, but it's really covering the main transgenic animal models, the type of mutations they show the promoter region they're covering and they're affected by, and so on. And however, what it's pretty clear is that all these models are useful for the familial Alzheimer's disease. There are also other mice that can be used to study the late onset Alzheimer's disease. In particular, I would mention here the SAMP8, which is the senescent accelerated mouse model. It has been partially useful to understand some of the mechanism and behavioral components of late onset Alzheimer's disease. However, what is known is that the genes that are responsible for the senescent process in the mouse are actually not very well known, or I would say are largely unknown. And another important component here, another important information is that there are nowadays no studies that have reported on the efficacy of currently available drugs for human beings in this specific SAMP8, this senescent accelerated mouse model. Additionally, another, let's say, concerning fact is that the pathological granules that have been identified in the brain of these SAMP8 senescent accelerated mice, in the beginning people were thinking, okay, these are the amyloids, so it's perfect. These mice are actually developing amyloids in their brain. However, it seems to be a false positive result. And this false positive result is due to a specific type of antibody reactions that you can see also depicted in this picture. So simply, the granules that we're observing in the brain of these SAMP8 mice are not beta amyloid plaques, but actually glycosidic neoepitope, identified by IgM antibodies. So what are we really looking at? That's a big question. Another point here that I want to make is that there is a bias. We cannot ignore this fact. There is a bias both in the peer review process in publication, but also in the peer review process of grant evaluation. Just to give you an example, this is an analysis that we have done looking at all the NIH, the United States National Institutes of Health, project that have been financially supported by the NIH in fiscal year 2007, 8, 9, and so on until 2014. And as you can see from the red bars, the number of projects and the total funded provided by the NIH supporting animal model-related studies is much, much higher than the number of studies focusing on either stem, patient-derived cells from patients, so talking about in vitro methods, or epidemiological studies in humans, or neuroimaging studies focusing again on human patients. This is definitely a disproportion. So a take-home message would be that probably animal models have been useful, yes, to recap and to summarize and to understand some of the cellular and molecular mechanisms of tauopathies, but also amyloid formation in the brain. However, it's pretty clear they're not helping us to predict efficacy of drugs for human beings. Let's also consider another thing, and this has been really very nicely covered by previous speakers before me. Alzheimer's disease is not simply a neurodegenerative syndrome. It is really, really entailed and much more complex than how it was envisioned before. There are several lifestyle factors that play a big, big role, and animal models are not helping us to depict all these lifestyle components. And when we talk about lifestyle factors, not just advanced age, as we know, not just diet, but for sure level of physical activity, a reduced cognitive stimulation. We heard tons of information also this morning that were really, really interesting. And the low socioeconomic status and low educational attainment, also poor sleep quality is strongly implicated. So sleep apnea, poor sleep quality in general seems to be correlated with a higher risk of Alzheimer's. Air pollution, so living in urban areas rather than the countryside, for instance, and smoking, or either the intake of metals, aluminum, for instance, or pesticide, insecticides. And we should, of course, mention risk factors correlated to metabolism, so metabolic syndromes in general, diabetes type 2, but also diabetes type 1. They're all implicated and strongly correlated with the higher risk of Alzheimer's. And again, all these factors seem to play a role, in particular, for the late onset Alzheimer's disease, which again, let's think about it. More than 95% of people affected by Alzheimer's are actually affected by the late onset form. This paper came out just this month, actually last month, August. It was published in Lancet Neurology, and it's kind of supporting what I have been talking about right now. So they're saying that it's true. In the United States, there is this kind of perspective and this type of trend that is suggesting that the increase of dementia is actually amazingly high, and it's expected to triple by 2050. However, some observational studies that have been conducted in five different studies, but conducted in 4 different European Western countries, in particular Sweden, the Netherlands, UK, and Spain, are showing that actually in the last 20, 30 years, the rate of dementia has not increased. It's stabilized. Why? Because lifestyle factors are playing a role. People are changing their lifestyle since when they're young. And of course, improving an optimum, as it's highlighted there in light blue, and a very strong and focused attention on preserving optimum health in early life plays a big role in preventing dementia. So this has to be taken into account. However, an important fact, how much are we investing in prevention? Not really that much, honestly. So if we look at the trend, this is a publication that came out from Kalitz and collaborators in the American Journal of Preventive Medicine 2015. And it's showing basically that in particular focusing on the fiscal year 2010 and fiscal year 2012, the total funding that the National Institute on Aging, NIA, it's one of the several institutes at the National Institute of Health, the total funding that the NIA has provided on prevention is really low. It's only 3.4% of the total budget. And also, another fact, as I was saying, late onset is the most prevalent form of Alzheimer's disease. However, one of our analyses, and we're going to publish these results soon, I hope, if you compare the number of NIH projects focused on the familial Alzheimer's disease, which is only 1.5% of total cases, it's much higher compared to the total number of projects that are focusing on the late onset. And this is, to our opinion, a strong disproportion. This doesn't make any sense. What's the point of focusing much more on the familial type, considering that the late onset is actually the one that is the highest, the most prevalent? Okay, so Big question. How can we tackle these issues? How can we solve the current financial situation? How can we improve current research? We can all agree that it's important to keep on funneling money into research. It's pretty clear. Everybody's advocating for let's put money into research. It's true. But we should also start thinking about and reconsidering what are we investing for? What are we paying for as a taxpayers? Does it make sense to keep on funding research the way it was funded so far up to now? Probably not. We should just reconsider priorities and strategies. This is an example. So in the last, I would say, 10, 15 years, in the toxicology and regulatory testing field, there have been a very strong need for, many people and many research institutions, but also the European Commission, and also in the United States, many research bodies are really advocating for a change, for a strong paradigm shift in the way research is conducted. This specific report came out in 2007 from the National Academy of Science. The title is Toxicity Testing in the 21st Century, a Vision and a Strategy. What this guide was talking about was the need to really implement human relevance in the way toxicological testing are conducted nowadays. They also introduced in the guide the so-called adverse outcome pathway concept. I will go into this in a second. But the main message of the guide was really to highlight the need to start focusing on human relevant methods and models rather than animals. This is an example of a publication that came out in 2014 showing how the so-called AOP, which stands for Adverse Outcome Pathway concept, can be implemented. So when we start looking at specific chemicals and the effect that a chemical might have, we should really start covering different levels of biological complexity to understand how the chemical works. And it's much more complex and will give you much more insights on the activity of a given chemical than just jabbing a needle in a mouse.
Attention please, attention please. At this time, we are conducting a required fire drill. I repeat, this is a drill. Attention please, attention please. At this time, we are conducting a required fire drill. I repeat, this is a drill.
Thank you. Okay, so understanding how the chemical really works. So injecting a chemical through different type of administration routes. It might be through injection, through inhalation, skin sensitization, and so on, into a mouse or any animal will not definitely provide that many information. You might observe some cellular pathological effect, but really digging into the molecular mechanism, epigenetic mechanism, genetic effect, it might be much more complicated. And there are really much better systems nowadays to start looking at this effect. So the question is, can we apply the same translational, So can we apply the adverse outcome pathway concept also in Alzheimer's disease research? The question would be, I think it's over. Okay. Yeah! Great! Congratulations, Hotel Marriott. Okay. Thank you. So, going back to the concept. If we apply the adverse outcome pathway concept and we try to describe, no. Okay. Yes, let's go. How can we apply this concept to the onset of Alzheimer's disease? And another question would be, how can we link environmental genetic causes with old person outcomes? Understanding what are the the signaling pathways that get perturbed at the onset of Alzheimer's and during the progression of Alzheimer's disease. And how can we also identify all these mechanisms looking at the multiple level of complexity, of biological complexity? So when we talk about multiple level of complexity, we should consider the population level, the organism, individual body level, the organ tissue level, the cell level, then going into the cells, the protein and signaling pathway level, up to the epigenetic and genetic level. What are the available tools that might be useful to cover all these different levels of complexity, trying to improve human relevance in particular? So let's think about the population and the organism level. We know that's pretty obvious. Observational, epidemiological, large-scale intervention studies on human beings is the best tool to use. However, just to remind ourselves, Kalitz, in his publication in 2015, have shown that the 3.4% of the National Institute of Aging is focusing on prevention, and 9% of total NIH funds are actually allotted to these studies. This is unacceptable. It's just too low. What shall I do? I don't know. Shall we have a break and just finish afterwards? Yeah, because it's impossible to talk. We can have lunch, have a break, and come back. Yeah. Okay. Yeah, no, indeed. Now it seems to be stopped, but if you can, we can really have a break and then come back, as you prefer. Shall I go ahead? Okay, yeah. Okay, so let's think about the body and tissue and organism level of complexity. Again, in this case, blood samples, cerebrospinal fluid samples, post-mortem brain samples are definitely the ideal tools. Again, human relevant tools. But we should also think about neuroimaging technologies. MRI, PET, connectomics are also helping to really understand human relevance in relation to Alzheimer's disease.
Attention, please.
No.
Attention, please. This concludes our fire drill. Thank you for your cooperation.
Oh, great. Yay.
Fire drill. Thank you for your cooperation.
Thank you for your cooperation. Okay. And then, again, thinking about the individual level, cognitive tests of any kind. Here I'm just talking about the MMC, the HBLT. There are much more complex tests that can be applied, as we know. And let's talk about the organ and tissue levels. So I don't know how many of you people are familiar with the use of human-induced pluripotent stem cells. So these cells are really powerful tools because they can be derived directly from either healthy controls or people affected by dementia. What you need to do is just take a simple blood, like 1 cc of blood, or either skin cells. These cells from the person affected by dementia, for instance, can be reprogrammed into stem cells through a process that is a little bit complicated, and I will not really start describing how cells can be reprogrammed. But they can become stem cells, and these stem cells can be further differentiated into many different cell types. And of course, thinking about Alzheimer's, they can be also differentiated into astrocytes and microglia and neuronal cells, providing really insights, information regarding how the pathology got developed and how new treatments can be tested also, accounting for really also patient complexity. So we can potentially take cells from many different patients affected by harboring different isoforms of specific alleles, and also trying to figure out how specific drugs, the same drug, can have different effects as considering the person and the patient. These cells can be also grown in much more complex systems. Nowadays, there are really many initiatives ongoing, both in the US, but also in Europe and in Asia, trying to improve the physiological relevance of these cellular models. They can be grown in three-dimensional systems with scaffold and matrix, recreating really the brain in vitro in a way. And they are commonly called also disease-in-a-dish models, and in particular Alzheimer-in-a-dish models. There are really many publications addressed at this point. Other more sophisticated technology that can be used to, again, resemble the complexity of physiological brain and tissues in general in vitro is the so-called microfluidic organ-on-a-chip technologies. Several institutes, like the Wyss Institutes at the Harvard University, or either in the US, but also in Europe, many other industrial corporations and university institutes are really developing these brand new technologies to start having much more rapid and reproducible sensitive platforms for drug testing. And it's quite interesting that some companies like Mimetas.com, as you can see there, in the Netherlands, are actually developing high throughput format of these specific tools to test different drugs at different concentration and time of exposure. Then, of course, when we start talking about protein, signaling pathways, gene, epigenetics, we should mention omics technologies. So when we talk about omics technologies, we can think about the multiplex fluorescence in situ visualization for DNA, RNA, proteins, microRNA, piRNA, whatever you can think about. And all this type of information coming from omics can be integrated into the so-called systems biology approaches and system pharmacology. What can we get out of all this type of tools and this type of data? So we can start thinking about and really realizing, defining what are the Alzheimer's disease-related pathways that get perturbed at the onset and during the progression of the disease. We can start defining drug targets, their efficacy and their toxicity in human relevant systems. We can also start having a little bit of data integration. And this data can be done, for instance, by integrating and interpreting big data coming from omics technologies, so genomics, metabolomics, lipidomics, together with patient memory scores and neuroimaging technologies. Why not? We have recently published a paper, a commentary in the Journal of Alzheimer's Disease that just came out recently, like a month ago. And this is basically the main picture, summarizing how all these technologies covering different levels of biological complexity can be pulled together to really have a snapshot, a much better, I would say, much better and broader overview of how Alzheimer's causation is working. What are the advantages of applying a human-based paradigm shift in Alzheimer's disease research? So, thinking about some of the advantages, for sure, a system-based understanding of complex human disease is the first one. A second advantage would be the possibility to use cost-effective and predictive data. Sometimes using animal models, as you know, might be very expensive and time-consuming. We can also think about discovering novel and multiple drug targets, specifically addressing and specifically focusing on Alzheimer's disease and human patients. We can also think about gathering human-relevant information earlier in the drug development process, which is very, very time-consuming and very expensive, with very, very few successes. And we can also think about reducing the so-called late-stage drug attrition, this so-called clinical failure rate I was talking about in the very beginning, 99.6% in Alzheimer's disease. What are the barriers? What are the challenges to overcome to really promote this paradigm shift? We have envisioned both technical and knowledge-related challenges. So let's think about the technical ones. So first of all, for sure, we should start characterizing a little bit more and validating many of these human-based methods. We should also start providing to any researchers who might have the will to start focusing on this type of tools. We should start providing better quality post-mortem tissues. Sometimes these post-mortem tissues are not of high quality at all. Another thing to consider is the need to further develop some of the imaging and computational technologies that are currently available, and also start improving a little bit more these platforms like computational approaches and simulatory approaches to really gather reliable data, gathering insights into in vitro and in vivo extrapolations. From the knowledge standpoint, what we need to do is to really propose new regulatory approaches. We need to persuade industry, funders, regulators, people at the NIH, and also scientists in general of a need for a change, a need to address human relevance a little bit more than how it's been done so far up to now. And also, we need to start harmonizing all these new tools that have been developed. So far, many of these tools have been developed by industries, and of course, they need to become available to any scientist, any at large, I would say. I just want to finish by providing a list of possible recommendations that we might think about to really improve the current situation. One possible recommendation would be to start implementing funding for the production and the centralized distribution of these patient-derived cells. They should be widely available to scientists working in the field, and they should not be that expensive. A second recommendation would be to start really allocating funding on research proposals presented at the NIH, but also to any other research funding bodies that are aiming to define and validate early biomarkers of Alzheimer's disease. Early diagnosis is very important. We should also start allocating funding on projects that are focusing on the highest, the most prevalent form of Alzheimer's disease, which is the late onset, rather than the familiar. It's important to focus on the familiar, but really there are tons of research that have been conducted so far and just very few efforts to start highlighting a little bit more the pathological mechanism of late onset Alzheimer's. We should also start allocating funding on NIH-funded centers that are conducting omics technologies, addressing really these omics type of information that can be gathered. And they might be really, really useful to start understanding complex interaction, lifestyle factors, how these lifestyle factors can impact epigenetics in human beings, nutrigenomics, either thinking about this. And also, the NIH probably should start creating specific RFA. RFA is an acronym, not maybe all of you are familiar with this, but it's a request for applications that should be really more focused on human-based research. And of course, we should start focusing and addressing and funneling money in prevention. So this is my last slide. I think to my vision and also to the PCRM vision I'm representing here, it's probably time is really probably ripe for a paradigm change, for a paradigm shift in the way things have been done so far. We should start really addressing human relevance a little bit more. And thank you for your attention. I'm sorry for all this beeping. And so if you have any question, let me know. Or if you want to ask me afterwards and you want to go for lunch, let me know. Whatever you prefer. Yes? I can't hear very well. Yeah. In your approach, do any of the models play any role then anymore or not? Now you mentioned all these human cell lines and so on. And so you don't need it, or will it play any role for mice? Yeah, I started with it. So I think you can hear me, right?
Okay, I just repeat the question shortly. In the model or in the approach, there is, as far as I understood it now, cells and so are deriving from humans. So you want to focus on human data, and are there still any roles for animal models or not? Like I don't know, mice, mouse, transgenic stuff, and so on.
So I think animal models have been honestly overused. That's the main point. So animal models have been useful to provide data, for sure. We have published thousands of publications addressing transgenic animals, how to treat and tweak these specific genes and how to see possible effects. I don't want to say that animals have not provided insights into the disease mechanism. This would be really untrue, honestly. But at the same time, if we really want to start focusing on what's occurring in human beings, why human beings, especially the late onset people, are developing dementia, what are the lifestyle components, we should consider the interspecies differences. We cannot ignore them. We think about nutritional intervention either in mice and rats, but mice and rats have different type of diets. They do not eat exactly like humans. They have also different gut microbiome, you know, the gut microbiota. So this is also another component that should be taken into account. And just to cite some of these examples, but also their immune system is particularly different. Their, the brain, the anatomy of the brain is different. The metabolism in general is different. So all these differences are actually the main responsible of the clinical failure rate we are facing. And this is really not a PCRM, not the Physicians Committee for Responsible Medicine perspective. It's a global, it's a worldwide concern that is really taken up by European Commission, by the NIH, because at the NIH, there is an institute that is called NCATS. And NCATS, NCATS, is really focusing on addressing novel alternative methods. So people, scientists in general, I would say, are aware that we need a paradigm change. So we have used animals, any type of animals, mainly rodents or rats and mice for the highest majority. They have been partially useful for sure, but I think it's important now to start addressing human relevance. And we have the tools. So this is important to mention. I think we should also start improving education at the universities. I mean, many young medical students, young PhD, undergrad students are not really aware of the new methodologies that can be used. They're not really trained that much in the use of induced pluripotent stem cells, these brand new technologies, the use of organ-on-a-chips. There are not that many information, and this information should be provided a little bit more than how it's been done so far. That's my opinion. Any question? Any other question? Okay, thank you for your attention.